mediator high and mediator low profiles of inflammation in acute Lyme disease. Now, of course, the question is, what happens to them after they've been treated? Now, I'm skipping to Clemens Struley's data because I want to show this first, but he had a very similar study design where he looked at acute treatment levels at time, so time 0 over 12 months, and he looked at 3 inflammatory mediators and showed that of the 3, the first 2, as you'd expect, acute disease, inflammatory mediators high, treatment, so 2-month follow-up, drop down to normal, okay? But one, IL-23, was high at acute infection, but never drops down to normal over 12 months. So this is the first evidence that there could be inflammatory markers, mediators that help subset out people with acute Lyme disease. These are treated patients that don't recover, right? Because if you have a post-treatment test for IL-23 at month 2 and it's still high, that's a group that's destined to have persistent symptoms, that's destined to have post-treatment Lyme disease. So Clemens' group showed that with Alan Steer for IL-23, and about the same time we showed it for CCL19, were the people that were destined to develop post-treatment Lyme disease, again, high at visit 1, but only the people in green that were going to develop post-treatment Lyme disease, the mediator stayed high. The people that were destined to return to health in purple and orange, the mediator level of CCL19 came down at their first follow-up visit. So in fact, at the first follow-up visit, if your CCL19 was elevated, you had a 14-fold higher risk of developing post-treatment Lyme disease 6 months later. So in essence, it served as a biomarker of people that were at risk for developing post-treatment Lyme disease 6 months later, where if it came down to normal at your first follow-up visit, you weren't going to develop post-treatment Lyme disease syndrome. So that offers an option for an intervention, right? Because you can do a follow-up visit and say, hey, your inflammatory mediator here is still elevated, maybe we need to do something, and not just watch for the next 6 months. And then Clemens' group, again, just published in the last few months these are European neuroborreliosis patients from Europe, and published again another mediator with interferon alpha elevated again in the people in dark that were going to develop post-treatment Lyme disease versus those that returned to health. This mediator was elevated. So we're starting to develop some really interesting inflammatory markers that could serve as biomarkers to stratify who's at risk of developing post-treatment Lyme disease. There's also autoantibodies that we've discovered. PAD2, which is an autoantibody that's important in MS. We were actually using the post-treatment Lyme patients as a control group, and they had higher PAD autoantibodies than the MS patients did, actually. And so we've started to discover new biomarkers around autoantibodies for post-treatment Lyme disease. I'm going to transition now to metabolomics. The study of the metabolome looks at small molecules. So these are fatty acids, lipids, things that are very small molecules that are identified on mass spec. This is a principal component analysis looking at healthy versus Lyme disease. And you can see that the healthy fingerprint of metabolic small molecules is separated in 2 dimensions from the Lyme patients. And so this was work done with John Belisle's group and using our samples. And it showed that the metabolome, and you've heard that throughout the day today, right? The metabolism is different in chronic fatigue and other illnesses. And this relates to energy metabolism and other types of metabolism. So this is actually, if you design this as a diagnostic test now, the metabolism fingerprint for people that are going to go on and develop post-treatment Lyme disease, defined 6 months later, right, because it's a chronic 6-month illness, is different than those that are going to return to health. And that pattern stays the same at 6 months and even out to a year. So the metabolism fingerprint of post-treatment Lyme disease is different than those that return to health after treatment of acute Lyme disease. So another potential diagnostic test, right? If you have this metabolome fingerprint of PTLD, you're destined to go on to have it a year later and to have PTLD. And this is work that's ongoing. It's also been used to differentiate Lyme disease from STARI, Southern Tick Associated. Next thing is a really exciting project. I don't have data yet, but it's so exciting I have to tell you about it anyway. This is work that Kristen Honey facilitated at HHS. We're working with DARPA on their ECHO project. This is the study of the epigenetics. So now we're talking about host response, but at the very, very proximal end of host response, we're talking about the DNA that's being modified in its chromatin accessibility, histone modification, methylation. So these are modifications of the DNA that occur in response to environmental insults, including infectious diseases, that change the DNA. Remember, when I went to medical school, we were taught DNA can't change, right? Well, they rewrote the rules. So DNA can change in response to infectious diseases, and then that changed DNA changes the RNA expression and protein synthesis. So Dr. Chu is going to tell you about RNA expression, gene expression later, but I'm telling you right now, we've taken it a step backwards and are looking for the patterns of DNA modification that predict those RNA expression levels. This is really cool stuff. It's done by Mount Sinai collaborators and Duke. They're doing, this is sponsored by DARPA, right? So they're looking at defense threats. So they can find things, that are infectious diseases, but they can also find things, like organophosphates, explosions, radiations, any environmental thing that impacts you changes your DNA epigenetics. And so the cool thing is we got them to work on Lyme disease. So right now there's a project looking at the impact of Borrelia burgdorferi on the epigenetic changes that could, the goal, actually DARPA is all about getting results. The goal is to have a diagnostic test for this like quickly based on this epigenetic project. And then I'm going to finish with one of my favorite things, which is CNS imaging, which again can act as a diagnostic. We've heard about CNS inflammation. This is an example of TSPO PET imaging. So this is TSPO PET imaging in a chronic Lyme patient compared to a control. This is the same kind of imaging that was done to document the NFL football player brain injury. So this is TSPO PET, and you can see that the red, again, as usual, is hot, is high. So this is a marker. TSPO binds glial cells that are activated and inflamed. And you can see that the brains of chronic Lyme patients have more microglial activation compared to the controls. And this is presumably the hypothesis, a marker of inflammation of glial cells in the chronic Lyme, persistent Lyme state, and shows that, again, inflammation in your brain is bad. Okay, so we want to know, well, why is it bad? So on neurocognitive testing, these patients have diminished processing speed and other measures of cognitive processing. And then we put, MRIs don't work very well in post-treatment Lyme patients, but functional MRIs, which you heard about earlier today, actually work really well because a functional MRI is like a stress test. So I won't give you your gambling thing I thought was cool for your stress test for your functional MRI. Ours isn't so fun. We just ask people to remember a few letters. And so you remember the two letters. I'll do it really quickly. Remember H and P, all right? What are those letters? Now give me the two letters after that in the alphabet. Two letters after that. It's hard, right? I can't do it. So that's the stress test in the fMRI scanner when you're doing a functional MRI. And we found a profoundly unanticipated result in functional MRI. The people with persistent Lyme, we've hypothesized that their gray matter would have to work harder, like you all were working harder to try to find the answer. Well, it turned out their gray matter didn't work as hard. It was hypometabolic, right? This theme keeps reemerging of actually diminished metabolism probably, because the fMRI measures blood flow, the imaging. So what we found is, there's probably only one person in the room that can read these, but this is actually the white matter showed it up on bold imaging. Well, functional MRI people say, well, that never happens. White matter, right? White matter is not supposed to show up on bold imaging. it means that the activations were occurring in white matter as if the white matter is trying to work harder, right? White matter is the wiring that connects all the pieces of your brain, and the white matter was going nuts. These are bold increases, and these are white matter tracks. We mapped them back to the actual white matter tracks, and they're activated, which we didn't expect to see. And so we're now pursuing this with a DoD grant. And white matter imaging is not hard to do. We're doing DTI imaging now. This is going to be a diagnostic test very quickly if this pattern of white matter activation, which goes along with the brain fog of post-treatment Lyme, persistent Lyme, if it pans out in this DoD study, DTI scans of white matter, you can do those right now. And nobody's ever really looked at this in post-treatment Lyme. So I'm really excited about this as also a potential diagnostic test. So looks like I got 4 minutes left. Wow. So action items, improved diagnostics are urgently needed for diagnosis of acute Lyme, but then also this infection-associated chronic illnesses associated with Lyme. The direct diagnostics for microbial nucleic acid and proteins are promising alternatives for serologic tests, and an even more exciting measurement of host responses, including inflammatory processes, gene regulation, metabolic changes, epigenetic signatures, and imaging are future opportunities for diagnosis. This is our team at Hopkins. It's a wonderful collaboration among multiple schools at Hopkins, and I thank you for the opportunity to share what we're doing. Thanks.
Okay, so one thing, we have a timer here for the rest of the speakers. Once you go to 0, then it starts kicking up the time. So John, when you thought you had 4 minutes left, you're actually 4 minutes over time. Oh, that's okay, but it was a great talk. It was well worth it. All right. Okay, well, our next speaker is another star. So Alberto Ashiro is a professor of epidemiology and nutrition at the Harvard School of Public Health, as well as professor of medicine at the Harvard Medical School. His work has focused on uncovering the causes of neurodegenerative diseases. He has a particular passion for ALS, but his most recent discovery has really made him famous, and that is the link between Epstein-Barr virus and multiple sclerosis. So we're really looking forward to your presentation again today, Alberto.
Hello, everyone. Thank you, Avi, and thank you for the invitation to be part of this workshop. See if I can share my screen, as I did in the. Okay, I think you should be seeing my screen at this point. Let me start with a more general statement, which may not fit exactly into the biomarker topic, but I think it's important to address it directly. And the question of causality. This requires a demonstration that the disease risk in people who are infected is greater than that in the uninfected, which seems an obvious statement, but it's often, if not forgotten, it's not often complied with. Because it's a very simple concept, but in many situations it can be difficult. Because this requires rigorous longitudinal studies. We repeated, in most cases, we repeated serum sample to determine the time of infection and the time of the biomarkers of interest. Just to give you an example, based on the previous discussion, we can see an elevation of inflammatory biomarkers, say IL-6 or CRP, in individual with long COVID higher than individual without long COVID. But we don't know whether that biomarker was elevated due to COVID infection, or whether people who already had the pre-existing higher inflammatory state were more likely to develop long COVID. So this, now let me go back to the topic you heard before, which is Epstein-Barr virus and MS. And then going a bit a bit back in time, just to, this was the general concept before the latest study. There was this impression that EBV negative individual had a very low risk of MACE, but this was based on case control and retrospective investigation. And then individual with mononucleosis had an increased risk of MACE. So when you go from the middle point being EBV negative to the right column being EBV positive with history of mononucleosis, your risk of MACE seems to jump up 30 or 40-fold compared to individuals that were infected during childhood. So I will mention very briefly the recent study that has been also covered before. But the purpose of the study was just to demonstrate that if you follow a large population, this was over 10 million people, and you look at those individuals who are EBV negative, can we really demonstrate that those individuals will not get MACE unless they're first infected with EBV? And that is what the study did. So about 6.8% of the individuals were EBV negative. This is a proportion you would expect among young adults in the US. And as expected, there was high infection rate during the 20 years or so, the duration of the study, an average of 10 years of follow-up for each individual. But MACE only basically occur in individuals who seroconverted for EBV. And in terms of the hazard ratio, relative risk is shown on the left. Following EBV infection, the risk of MACE was 32-fold higher. And contrary to what was said in the first lecture, actually the risk in those individuals who were already positive at baseline was 26-fold higher. There's been that unfortunate paper of Horwitz et al. Who misinterpreted our data, and we tried to clarify, was clarified in an editorial in the MS journal. That was just a mistake of the authors. So the risk in individuals who were already infected with EBV is quite close to those who seroconverted during the follow-up. And importantly, as you see on the right, being this in an observational study, we needed to get rigorous controls. And we use CMV infection as a negative control, meaning not expected to increase the risk of MACE. And as you see there, seroconversion for CMV was not associated with MACE risk. And those individuals already positive for CMV had in fact a reduced risk of MACE. An observation that has been reported before, and you heard about CMV in previous talk. In terms of temporality, it was important to demonstrate that EBV infection preceded not only the first symptoms of MACE, but the earliest sign of the disease. So we measured neurofilament, serum neurofilament light chain levels that is shown here were already elevated 6 years before the onset of symptoms. And we demonstrated, as you look on the left, that these are within persons serum NFL level, that serum NFL remain constant around the time of EBV infection and increase only after EBV infection. So EBV infection occurred at some point between the first and the second green dot on the left. And shown on the right, EBV infection by itself in individual who didn't develop MACE does not cause an increase in serum NFL. So this shows very clearly that the EBV infection precedes serum NFL elevation. And then finally, in terms of the specificity, looking at several other viruses that could potentially trigger MACE, and we found that this was a screening using VIRScan, covering over 100,000 peptide, 56 amino acid overlapping by 28, showing that all the peptide enriched among the MACE cases, both before and after the onset of MACE, were EBV peptide. Conclusions are listed here. So MACE is a rare complication of EBV infection, which of course does not exclude the role of other factors, and the main factors that we and others investigated over time are listed here. Now let me focus more on the question of biomarkers. So from here on, I'm talking about people who are EBV positive, and I am looking at blood sample collected before the onset of the first neurological symptoms of MACE. We started this study over 20 years ago. It's easier to study antibody titers than to study EBV negativity, just because EBV negativity is so rare. So this is our first study looking at different antibody against EBV, and CMV always as a negative control, in serum sample collected just 2 years before, a median of 2 years before the onset of neurological symptoms. And this study were done using indirect immunofluorescence that now is difficult to find a lab doing it anymore for EBV serology. As you see here, we had a signal for EBNA complex, EBNA, which is a combination of all the latent nuclear antigen, EBNA1, EBNA2, no signal for VCA in this first study, and a signal for the early antigen. Then we follow up with a second study in which independent study, the first was in a population of female nurses in the US. This is already the military population, but one of the first subset with 80 cases. And you can see here the strongest signal is for EBNA complex. There is still a signal for EBNA1. EBNA2 becomes sort of weaker, and the early antigen is still present. But when you look at the individual with case collected at least 5 years before the onset of symptoms, see that the strongest signal seem to converge to EBNA complex and EBNA1. And again, looking in a different cohort, this is in Finland, and looking prospectively. And at this point, we started focusing on EBNA1 for two reasons. One, that nobody was doing immunofluorescence for the EBNA complex anymore. And second, because this seemed to be at least one of the strongest antigen. And in particular, want to spend a few seconds on this at least. If you look at the top left, this is again the military population. All of the individuals are EBV positive. The serum level of anti-EBNA complex titers are strongly monotonically associated with an increased risk of MS, going up to 36-fold. At this stage, we're still measuring anti-EBNA1, as you see on the right. And this is a question, I'm not an immunologist, but it's a question, for those of you in the audience who are more immunologists or virologists, is this a chance? Or are we looking really, there is something that this signal against EBNA complex, could that be even stronger than the EBNA1 titer that have you seen before? Is the one in which all the molecular mimicry work has been focusing. EBNA2, antibodies against EBNA2 have become less strong in this population, and anti-VCA are also all significant, but not as compelling as the anti-EBNA complex and anti-EBNA1 titers. What does this tell us? So the one interpretation is the molecular mimicry that you heard before, but there is also the possibility that this high anti-EBNA titer represents something different. And this whole study, this was done among atomic bomb survivors of all the population in Japan. And the reason I'm choosing this because I didn't really find other study correlating the anti-EBNA complex antibody titers with the precursor frequency of cytotoxic T cell against EBV. So at least according to this study, anti-EBNA titers may be a marker of a strong cellular immune response against EBV. So this going parallel could complement the hypothesis of the antibody molecular mimicry. Now, the association with anti-EBNA is so strong that you may wonder whether it could be used for diagnostic purposes. I don't really think so. This is ROC showing the sensitivity versus specificity of antibodies against the EBNA complex. So even if the association is very strong, EBNA complex on the top, EBNA1 on the bottom, still in terms of prediction, I'm not sure it's sufficiently useful. Another aspect of these antibodies is that elevated levels are seen up to 16 to 20 years before the onset of MS. And this is an early study that we did with the Kaiser Permanente population. The red line represents the anti-EBNA complex titers or anti-EBNA1 titers among the controls. And as you see, even 16 to 20 years before, they're both significantly elevated. This, suggests before our latest study already suggested that this should precede the onset of MS. Now, another puzzling feature of these antibodies is that they don't seem to predict the disease severity or progression. So this is a study in benefit. This was a trial of over 400 individuals with early MS. We will say today at the time was called clinically isolated syndrome. And if you look at the left, these are quartiles of anti-EBNA1 titers. So the highest quartile is shown in red, and this is the probability of survival, meaning probability of not developing clinical definite MS. So we are just looking at the progression of the diseases. You see, there is absolutely no evidence that having higher antibody titers is associated with faster disease progression. And the same is true for the anti-VCA. And previous study also have not found any convincing evidence that the titers of these antibodies predict the long-term progression of disease. So the question of the peptide, I think, has been well covered by Dr. Robinson. I just listed here the peptide in our study that are more strongly associated with MS risk. And you see on top is the EBNA1 peptide, and the sequence, the segment that is on top is the same. From 365 to 420 is the same sequence that is be found by the group in Stanford to cross-react with glial CAM, and this has been mentioned before. So finally, I wanted to mention this paper because I think we lack comparable data, prospective data, and will be difficult to get them on cellular immunity. Is there any way we can study the cellular immunity against EBV before the onset of MS? This is a study done in individuals with clinical MS comparing to controls, and this is shown here on the left. You have SARS-CoV-2, just showing that the frequency of cytotoxic CD8 T cells against SARS-CoV-2 is higher in individuals with COVID in green on the right of the left side of the figure than in controls. And comparably, in MS, the frequency of cytotoxic T cell against EBV is higher than in controls. So this is just an analogy showing that the magnitude of the difference between the two groups is comparable, consistent with the idea that EBV is not only causing MS, but is also driving the disease process. We have a blood sample, serum sample collected before the onset of MS and before the EBV infection in a large population. I wish I could present the data. We are trying to get funding to look at in depth at the sequence of event in terms of not only of anti-EBNA titer as I've shown here, but in terms of the specific peptide that are involved, and also the specific host peptides and protein, possibly to identify, Dr. Robinson mentioned 25% of his patients could be potentially explained by this cross-reactivity between EBNA1 and glial CAM, and it's likely that there could be other cross-reactivities that could explain other cases, if molecular mimicry turns out to be the main mechanism, which we are not sure yet. And this just to show that this is the same study that I just shown the previous slide, that not only there is a higher frequency of cytotoxic T cells against EBV in MACE, but this is shown in green in this pie at the bottom right corner of the slide tend to recognize lytic antigen, which is consistent with the idea that EBV reactivation in the brain is the leading cause of the pathology. And just recognizing here, for a question of time, just the main collaborators, but among many others who helped for the study. Thank you.
Thanks, Alberto, for that great talk. I have to say that I've heard you speak many times, but every time I learn something new and the association keeps getting stronger. Beautiful work. Thanks. Okay, the next speaker is Dr. Razia Pretorius, and she's a distinguished professor and chair of physiological science at Stellenbosch University and an honorary professor at Liverpool University. Her work has focused on early detection of inflammatory diseases and led to the discovery of microclots as a novel biomarker for long COVID. We appreciate your coming all the way from South Africa to give this talk. All right, thanks.
Hello, everyone. Thank you very much for the kind introduction, Avi. I also just like to thank everyone that has been listening online and in this meeting. I know it's been a long day, and for some it's been very late staying up. So today, I would just like to acknowledge some of the researchers and some of my students that have been working in my lab, particularly Professor Douglas B. Kell. He's my longtime collaborator from the University of Liverpool, and then various clinicians, and also a team of scientists and researchers and postgraduate students, Massimo Nunes, Tamasha, Simone, and then also Chantal Fenter, who is my physiologist and in our lab, in our blood lab, the blood lab manager. And then this work has been sponsored by various grants, and I will show that during this presentation. So the focus of this talk today, I would like to place a little bit of emphasis on the relevance of receptor and inflammatory marker interactions driving various disease pathologies. I would also like to discuss the relevance of direct protein-protein interactions, particularly in clotting pathologies, and the interactions between inflammatory molecules, also those from viral and bacterial origins, and how they interact with proteins, specifically the clotting protein fibrinogen. And then the question that I think we all have is there a place for novel methods in diagnosing well-known as well as new diseases? Over the years, my lab and my collaborators all over the world have been looking at how inflammatory molecules may cause pathological blood clotting. And as I mentioned, these molecules, these inflammagens, might be both viral and bacterial inflammagens. And we have been studying the effects of these circulating inflammatory molecules on platelets and red blood cells and the clotting protein fibrinogen. Platelets in circulation play a very important role, as we know. But one of the things that was quite interesting for us, and we have been focusing on, is the fact that platelets have numerous receptors on their membranes that can interact with and cause platelet hyperactivation when these inflammatory molecules are in circulation. Platelets also have got a significant role to play in endothelial cell signaling. And here are just some of the diagrams that we've been publishing on, where platelets have a direct interaction with endothelial cells as well as within cells. In the context of COVID-19, it's very important to keep in mind that platelets form complexes, not only with themselves, but also with immune cells. They can form numerous immune complexes that can drive autoimmunity, immunity, and all sorts of platelet hyperactivation interactions with other cells, the endothelial cells, as well as red blood cells. We also, as I mentioned, study clotting pathology. We have been looking at how in circulation, if there are inflammatory molecules that might cause direct protein-protein interactions by binding to the clotting protein fibrinogen. Fibrinogen is, when you are healthy, a soluble protein. However, in any type of inflammatory molecule in circulation, there can be a direct protein-protein interaction causing the fibrinogen to change its shape. And this shape changes we have shown to be an amyloid nature. So if anything binds to fibrinogen, it will cause various interaction property changes. If we look at a typical protein, a protein typically has got lots of alpha coils and little beta sheets. However, when there are interaction with inflammatory molecules, the alpha coils untwist into these large beta sheets, and that causes pathological clotting. And we have shown that a molecule that can directly interact with open hydrophobic areas on the protein, on the fibrinogen protein, where damage has occurred, namely THT, can show up these amyloid changes. So THT, or theoflavin T, is a well-known molecule for amyloid protein, and it has been used previously in conditions like Alzheimer's, where THT have shown amyloid protein changes in the brains of Alzheimer's individuals. So when these clotting pathologies happen and THT bind to these open hydrophobic areas, we can view the changes by looking at fluorescence microscopy, and we could also view it by looking at scanning electron microscopy, and that's what we have been doing the past years. However, in 2020 came along COVID-19. So we were well placed. We were looking at clotting pathologies all along. We had the correct tools. We had the patients in South Africa. And it soon became clear that in the cohort of patients in the acute stages right in 2020, that platelets were significantly hyperactivated. I point your attention to the yellow arrows, where platelets have seemingly been activated and interacting with themselves, as well as interacting with red blood cells in the middle panel. Those are scanning electron microscope micrographs. And we also soon found that not only are these platelets and red blood cells interacting with each other with severe pathology, we also found that there are significant microclot formation or deposits in samples from ICU patients. And you can even see that these microclots glue together red blood cells. Look at the blue arrows on the far micrographs. We then immediately also, at that stage, looked at the clotting protein. We took platelet-poor plasma and we exposed it to our cohort. We had healthy plasma, we had type 2 diabetes, and in the COVID plasma. And to our surprise, we found that in COVID plasma, there was significant microclot formation, much more than we have previously noted in type 2 diabetes and obviously also in controls. So the green signal is from theoflavin T, where it binds to damaged areas of the clotting protein, and these damaged areas then shows up as this microclot formation. We also looked at the platelets, and here you see some of the platelets. A is a typical control, where a platelet just has little micro structures where pseudopodia is developing, and that's due to contact activation. But in the blood of patients with acute COVID, numerous platelet activities can be seen. They are spread, they are connected to each other. We use two markers here, and this PAC1 as well as p-selectin marker. And the p-selectin is the marker for that shows up in pink here, and PAC1, the green marker. So those two markers are indicative of platelet hyperactivation. P-selectin, as you might know, is a very interesting molecule. It can either be inside the platelet, inside the alpha granules when the platelet is healthy, or it can travel onto the membrane when the alpha granules are activated, or it can be shed as soluble p-selectin. We looked at both. We looked at the soluble p-selectin, but we also then found these numerous platelet p-selectin places where it's found on the membrane, where it acts then as a receptor for binding platelets to each other and to the endothelial cells. After those initial studies looking at acute COVID, we also thought that let's see what's happening if we can use the spike protein. There is one part of the protein, and we exposed that to healthy plasma. And here you could see that in the experiments that we published in 2021, that spike protein can trigger significant platelet hyperactivation as well as microclot formation. And that's in line what some of our collaborators from Sweden has said, Sophie Nailstrom, where they found that the spike protein itself is an amyloidogenic protein. So it is interesting for us then at that stage in 2021 to see that the spike protein could indeed also trigger microclot and platelet hyperactivation. We also again looked at scanning electron microscopy, and we also looked at microfluidics. So here you can see the effects of the spike protein at very low levels, where we could actually trigger a microclot just by adding spike protein to a healthy individual that looked very much similar to COVID-19 microclots. And the COVID-19 microclots you could see in the middle there with the red arrow, and the bottom, the spike protein that triggered microclot formation in our microfluidic system. We also looked at the differences between microclot formation as well as platelet hyperactivation when we looked at healthy individuals, Omicron, as well as the earlier more virulent Beta and Delta variants. We all know that the Beta and the Delta and the earlier variants cause significant disease, where Omicron perhaps not so severe and so many deaths happening from the acute infection. What was quite interesting for us was that in Omicron, much less microclot formation were prevalent in the acute phase. But if we look at the platelet hyperactivation, we could not really see a difference between the various variants. Then obviously we turned our attention to long COVID, and that was probably the end of 2020, beginning of 2021. It soon became prevalent that there were numerous patients that did not recover fully from the acute phase, and where we still saw significant symptoms that persisted. And when we looked at the platelets, we saw significant platelet hyperactivation, platelets just clotting together and binding to each other, forming complexes with each other. Here you could see when we compare it to healthy platelets, there's simply no comparison between the level of platelet hyperactivation that we found in long COVID. Obviously, we were looking at that stage using a fluorescence microscopy method. So for diagnosis, it's quite a difficult methodology because very few pathology, in fact, we could not really find pathology labs at that stage that uses such instrumentation. So at that early stage, the earliest thing for us was to develop a platelet grading system to visually show how we see and how we anticipate a person with long COVID or acute COVID would look like. And here is our platelet grading system, where you could see the first row is how you would typically find healthy platelets. Perhaps the second and the third row, you would find platelet hyperactivation in someone with diabetes or rheumatoid arthritis. And the fourth row, you would see typically patients with long COVID and acute COVID. And then same for the clumping. No clumping in healthy individuals, we ever note. However, severe clumping in people with long COVID as well as people with acute COVID. Same with our microclot grading system. We developed that as well. And here you can see the various clotting pathologies that we note. Then we also looked at proteomics. So proteomics was interesting for us because we wanted to find out what is inside the clots. And to our surprise, we could not digest the microclots when we used a basic enzymatic process to digest them that you would use for mass spectrometry. When we first did our first digestion step, we could still find microclots in acute COVID and long COVID, but not in diabetes or in our control sample. When we could eventually digest all our microclots using a process that we published, we found numerous inflammatory molecules entrapped inside the clots. We found von Willebrand factor. Obviously, we will find our various fibrinogen molecules. But we found a molecule called alpha-2 antiplasmin, and that was quite interesting for us. Because if you look at the clotting diagram here, and if you look specifically at alpha-2 antiplasmin, it prevents clot breakdown. And that's what we have been seeing. Clot breakdown doesn't happen as it's supposed to. We repeated the study, the proteomics analysis in 2022 with a much larger cohort. And then we found the same results, but we also found some interesting other molecules. And also entrapped in these microclots, we found numerous antibodies as well as autoantibodies. So now the question was, what next? Do we keep on using our microscopy method, or should we rather focus on new methodologies? So here's a place for imaging flow cytometry that we thought would be a good one. And thanks to funders, Kernels, Balvi, as well as Poly Bioresearch Foundation, we could develop a method, a cell-free method to detect microclots inside plasma from patients with long COVID. And here is just some of our analysis. This is really very new. Our paper is currently under review, and we hope that this will be a methodology to be used by pathology labs. There's just some of our results where you could see the microclots are really significant, and one could detect it using a cell-free plasma model. If I just briefly turn my attention to ME/CFS, then ME/CFS, we recently also published together with Massimo Nunes, my PhD student, where we also found microclotting and platelet hyperactivation. Microclotting were not found to the extent that we were expecting to see in long COVID. So not the same, but there were still significant clotting pathologies. We just concluded a proteomic study where we also saw numerous clotting pathologies using proteomics of our ME/CFS cohort. We have just been doing, together with my students, some further results that I would like to show you here, where we stained the long COVID microclots for biofilm, and we used a biofilm tracer. And here you could see some of our results, where inside these microclots that do not break down during our proteomic study, and therefore where we found some of the molecules entrapped inside the microclots to prevent microclot breakdown, we also found some biofilm molecules inside these clots. And here we just did a study where we looked at long COVID samples stained for biofilm as well as amitracker. So amitracker is a replacement stain for theoflavin T that we used to also show amyloid changes inside the microclots, and they stain at a little bit different areas. What we also recently found is just very, very, very new for us. We're still looking at the data. We found endothelial debris in our platelet-poor plasma. We always found it and saw it, but we didn't confirm it yet. But recently, Massimo Nunes, my student, just confirmed that both in long COVID and in ME/CFS, these structures that we see definitely have an endothelial origin, and the endothelial origin we confirmed using the marker E-selectin, which is also a fluorescent marker. So in short, we have been finding ischemic reperfusion injury in all of our patients that we have looked at. Many people have shown that. So we think that if we look at everything that we have seen, all the endothelial damage, the microclotting, that the microclots that don't break down, the platelet hyperactivation, we can say that long COVID is possibly a thrombotic endothelitis. And just to quickly show, I'm not a clinician, so I'll just quickly show you some of our data from our clinician-initiated treatment regime, where 91 patients were treated with anticoagulants. We found that 80% of these patients were recovered, and that is quite nice information for us. And that was also determined by using a patient PGIC scoring. And when patients said they felt better, we could also not anymore find these huge microclots. So the microclot load would have decreased together with how patients felt. So in conclusion, there's definitely a relevance of receptor inflammatory marker interactions that drive disease. We need to look, when we look at diagnostics and new methodologies for diagnosis, to look at protein-protein interactions. And there's definitely a place for novel methodologies, including fluorescent microscopy, perhaps also imaging and normal flow cytometry, proteomics, although I know it's not necessarily an easy thing to do as a point of care. But then the question is there possibly a place for novel point of care technologies from a drop of blood? Thank you very much.
Laboratory medicine and the Department of Medicine at University of California in San Francisco. He's also the director of the UCSF Abbott Viral Diagnostics and Discovery Center. He currently leads the Translational Research Laboratory focused on metagenomics, next-generation sequencing assay development for infectious disease diagnostics and investigation of the pathogenesis of immune responses to emerging pathogens. He's also developing new technologies such as transcriptome profiling and predictive models using machine learning for host response-based diagnosis of infections. Thanks very much, and we're looking forward to your talk.
Great. Thank you, Avi, and I'd also like to thank the organizers for inviting me to speak with you today. My talk is titled Clinical Metagenomic and Host Response RNA Sequencing or Transcriptome Profiling for Diagnosis of Infection-Associated Chronic Illnesses. The prior speakers have already gone over kind of the various diagnostic modalities. In this talk, I'll focus exclusively on metagenomic sequencing. What is it and how we can apply it potentially for aiding in diagnoses, as well as transcriptome profiling or host response sequencing. Now, I do have some disclosures. I'm the inventor of software which analyzes data, metagenomic data for pathogen identification. I'm on the scientific advisory board and a co-founder of a few companies. So I'm an infectious disease doctor, and infectious diseases, at least for acute infectious diseases, we're faced with major diagnostic challenges. It's true that up to 62% of the time in cases of pneumonia in patients who present to the hospital, they're often admitted to intensive care unit. We can't diagnose an infection despite extensive diagnostic testing. When meningitis encephalitis or neurological illnesses, which are thought to be potentially infectious in etiology, about 50% of the time, the cause remains unknown. And even in perhaps well-characterized infectious syndromes such as sepsis, over 20% of the time, we can't make a diagnosis. And recent studies have suggested it's even more than that. And I think the failure to obtain a timely diagnosis of these infections leads to delayed and inappropriate therapy, increased mortality, and excess healthcare costs. And as a microbiologist and infectious disease physician, the traditional paradigm of microbiology is to identify the organism. Whether we want to look for DNA from the organism or RNA, whether we want to culture the organism or identify antibodies to the organism. And up until now, really the only major indirect tests we have are antibody detection as a way to diagnose infections. So my interest in my laboratory has been in developing clinical metagenomic sequencing as a way to diagnose acute illnesses. And I want to talk to you today about how potentially we can apply that to chronic illnesses. But I'm the director of the UCSF Clinical Microbiology Laboratory, and it's a highly regulated environment. So what we had to do is we not only had to develop this test in the research lab, but we had to move over to a clinical lab where it could be fully validated. We could evaluate performance characteristics. And a large part of metagenomic sequencing involves bioinformatics, analyzing the sequencing data. We're approaching a metagenomic sequencing approach, meaning that we're not using specific primers or probes. We're not targeting any one organism. The general concept behind metagenomic sequencing is you want to interrogate the metagenome. Just sequence all of the DNA, all of the RNA in clinical samples, and in principle, you should be able to identify any organism. Whether it's a virus, a bacterium, a fungus, or a parasite, as long as there is DNA and RNA from that organism. And so we want to develop that so it could be done as a routine clinical test. And this sort of gives you the idea, because nearly all pathogens, the one exception would probably be prions, which are protein-based, can be identified using metagenomic sequencing. And this is why it also becomes a very powerful tool for investigating emerging infections. We've actually shown kind of the potential clinical utility of this tool. We have several publications, both of the laboratory validation as well as the use of it in prospective clinical studies, comparing it head-to-head with all microbiological tests that are available. We've also been able to show that you can run the same assay on a variety of different body fluids. We began with spinal fluid for diagnosis of neurological infections, but we've been able to extend it essentially to every possible body fluid and tissues as well. And currently, we have what we can clinically validated clinical assay that we can run at UC San Francisco for doing metagenomic sequencing for identifying pathogens from spinal fluid, from plasma, as well as from various body fluids, such as respiratory fluid. One of the components of it, though, is that, and one of the challenges has always been, like, how do you actually analyze this data? How do you actually get results? And so for that, we actually developed in my laboratory an automated pipeline, which is currently now being run on the cloud. So essentially what it does is it pulls all of the raw sequence data, analyzes it in the cloud under a HIPAA-compliant pipeline, and then it produces results that are interpretable by a laboratory director. So importantly, you don't need to have any bioinformatics expertise. You can treat it as a black box about getting results directly from the clinical data. And so we've found by moving it to the cloud that it can directly address the computational analysis bottleneck that's imposed by it. I don't really have time to go over this particular study, but this was a study that we published in New England Journal of Medicine a few years ago, showing that head-to-head, that metagenomic sequencing can provide more diagnoses that you can get from when compared to extensive conventional clinical testing. Roughly one quarter of the infectious disease diagnoses had to be made using metagenomic sequencing alone, of which the majority of those had direct clinical impact, meaning that the results of the metagenomic sequencing test affected treatment or management of the patient. And I want to also show you this slide. This is in the roughly 7 years we've been running the test from spinal fluid for diagnosing neurological infections and also from plasma. You can see here that it really represents the full spectrum of organisms that can cause disease, including several of the organisms that are mentioned here, Borrelia, for instance. We've also been able to identify kind of emerging and unusual infections, including astrovirus infections of the CNS, cholera tick fever virus, unusual parasites, and bacteria that can actually cause disease. Okay. We've also been able to show that it works on different body fluids. I want to introduce this slide because we're currently working on ways that we can actually introduce it to the clinical lab. And the idea here is that you don't need to have a specialized reference lab to run this test. We want to make it so that pretty much any clinical lab can run the test. So we've managed to reduce the MNGS assay procedure from something which takes 2 to 3 days to something that you potentially could do in the same day. And we reduced it from 300 steps down to 20 steps, where the hands-on time is from roughly about 10 hours for a clinical laboratory scientist to run the test to about a 2-hour hands-on time where you can actually get this test. So I believe that it's going to be feasible in the near future that we can actually perform this analysis routinely as part of a clinical or public health lab investigation. I want to move now, and how does this all apply to chronic illness? Well, one of the interesting things about doing metagenomic sequencing is that we're sequencing all of the DNA and the RNA. And so that brings up, most of what we end up sequencing is actually not the pathogen. Usually the pathogen happens to be a very small fraction. Those are the non-human reads, typically less than 1% of the sequencing data. The vast majority of the sequencing data comes from the human DNA and RNA from the patient. And so as a result, what we're essentially doing is whenever we run a clinical metagenomic sequencing test to look for pathogens, we're actually doing an RNA-seq experiment. We're actually looking at the patient's host response, or we have the capability of generating that data. So the idea here is that why not simply repurpose the data that we're already generating for the purposes of pathogen detection, repurpose the RNA data to identify patterns of gene expression that can help us aid in diagnoses. And similarly, we've been able to show that you can look at, for instance, the DNA portion of it to be able to diagnose, for instance, cancer potentially, because we have several publications where you're looking at copy number variants. And in this way, we can sort of like really leverage all of the data that's coming from this assay. And so this basically reflects kind of the goal of implementing precision medicine in infectious diseases. Now, precision medicine is the overall concept is the application of omics technologies that allow us to basically personalize medicine or personalize the management or treatment of patients. In cancer, that's revolutionized when we realize that all cancers are different. And similarly, I think we have to change our thinking in that infections are different as well, and a patient's host response to infection in particular is different. And I think that's really important given the context of this particular particular conference, simply because we've been talking all about aberrant immune responses, aberrant host responses. We've been talking about persistent organ infections. And I would argue that the situation is probably different between different individuals. We know that the response to West Nile meningoencephalitis, some patients are asymptomatic, some patients have a fatal encephalitis. I mean, the same is true for SARS coronavirus 2. Many individuals can get infected but have no symptoms, whereas others develop a critical illness and pneumonia. And what I want to argue is that the host immune response or the host response to infection plays a critical role in that. So what I want to do here is, the goal is actually to leverage all this data, take the RNA sequencing data and use transcriptomics or gene expression profiling as a way to better develop diagnostic tests for chronic illnesses. And I want to introduce as a concept, the general concept is this: why not do transcriptome profiling, take the RNA data, do the transcriptome profiling, identify differentially expressed genes that can help you produce a differential diagnosis for different diseases. To give you an example, what you see on the lower right, those are the major disease-specific pathways associated with Lyme disease. And many of them we already know about. We know that TREM1 signaling is involved. We know that NF-kappa B signaling involved. We know that Lyme disease in some ways represents a viral infection because of toll-like receptor signaling. And so, but this is all data that's not generated from some targeted method. This is all coming directly from the RNA-seq data. So I didn't select these pathways. The machine learning algorithm selected these pathways as the ones that are important. And so in general, I want to let AI do everything. I mean, we know that AI is going to become our masters in a few years. So, and as you know from ChatGPT, so the idea here is, why not let AI actually do the analysis to allow you to identify, for instance, in this case, specific features or genes that are specific for a given disease. And similarly, this is how Google Image works, where you train the AI algorithm on images of cars. And by doing so, the idea is, by using 80% of the data for training, it can not only identify the cars that went into the training, but potentially it can identify similar cars. And so this is the general idea, in that we divide the data 80-20. We use 80% of the data to train the classifier, and then we use the remaining 20% to actually evaluate the classifier performance. So this is how it looks. It's a little more detailed than kind of the car analogy, but the general concept is the same. I now have data from roughly 1,000 spinal fluid samples, which are well characterized, representing bacterial infections, viral infections, fungal infections, parasitic infections, autoantibody syndromes, and other non-infectious etiologies. So why not simply take all of that, use a machine learning-based model to train, and then by doing so, you could then identify specific genes or panels of genes that can diagnose infections. And so we've actually done so. What we did is we divided these into roughly 4 major categories. So we have a category called AINI, which stands for autoimmune non-infectious etiology. That includes autoantibody syndromes, neurosarcoidosis, amyloidosis, lupus, we have PANS and PANDAS cases, et cetera. And so the idea is to lump them all together. And then we have specific categories of bacterial infection, including atypical bacteria such as Mycobacterium tuberculosis, fungal infection, and viral infection. And can the classifier then be able to distinguish between these? So this is called the multinomial classifier, in that there are 4 categories here. And we're able to show, as you can see here, the test set results are never as good as the training set, because the data is based on the training set model. But you can see here that we're able to obtain roughly a 90% accuracy or more for each of the 4 comparisons. And the idea right now is that we can add this as information that can be provided in the metagenomic test result. I also want to show you some other examples. We can actually achieve some information at a higher level of granularity. So what you see on the right are actually cases of acute fasciomyelitis in children. So these are all coming spinal fluid samples from children presenting with acute fasciomyelitis. This is a poliomyelitis-like syndrome. It typically is characterized by paralysis. It's also characterized by an inflammation of the gray matter of the spinal cord. And a hallmark of AFM, or what's well known about AFM, is that you can't really identify the enterovirus that appears to cause it, which is enterovirus D68 in spinal fluid. In fact, the diagnosis is always made essentially incidentally because we're able to identify enterovirus D68, for instance, in respiratory secretions, or perhaps in stool, or occasionally in blood, but you almost never identify from spinal fluid. And that's why it's been very difficult for us to establish the cause. It actually took several years for us to, now I believe the scientific consensus is that enterovirus D68 is certainly one of the causes of AFM. But you can see here that these are cases that are all microbiologically negative. So there are negative spinal fluid samples, but you can see here that we can actually identify a signature that appears to identify what the AFM signature looks like if it's associated with enterovirus A71 infection or D68 infection. And what's also quite interesting is you can see here that the classifier actually doesn't do a good job of identifying enterovirus meningitis. And probably because there are over 101 enteroviruses that cause meningitis, there probably is a lot of heterogeneity in the host response to enterovirus meningitis. But if you have a very specific disease syndrome, like acute flossamyelitis, it appears that the classifier does a pretty good job of that. Another example is where we can potentially identify specific types of infections. Let me just go back a slide, sorry. If you look on the left, you can see that we can do like what's called an LDA plot. It's similar to a PCA plot, but the general idea is that we can identify categories of infection. So we can use a classifier, for instance, to identify specifically Mycobacterium TB, and not simply to identify it as a bacterial infection. I also want to mention some other specific use cases. We've been able to identify, for instance, a classifier that can potentially diagnose cocci or histoplasma. These are two dimorphic mold infections that are commonly encountered in infectious diseases. And we can also identify parasitic infections. It does appear to also have a classification. And so what we might end up with is something like this, where in addition to a metagenomic sequencing result, we're going to be able to provide kind of a host response classifier that can identify not only specific types of infections that we can identify by direct detection, but also potentially non-infectious causes such as GABA receptor-mediated autoencephalitis, which is an autoantibody syndrome. You can see here by running the host response, it's not looking specifically for the GABA receptor or any sort of target with the GABA receptor. It's just looking for a signature that would suggest that it's not infectious, and it does appear to classify it correctly. So you might imagine that you have a kind of a spectrum of host response. And in this case, this is where I took all of the diseases, infectious diseases, non-infectious diseases together, and I simply essentially tried to sort of essentially use the algorithm to sort of place them or cluster them in an unsupervised fashion. And the idea here is that you can, at a glance, you can kind of see what we already knew. We knew, for instance, that autoimmune disease is closely tied in with viral infection. As you can see, there's quite a bit of overlap there. But we can also see that we can identify clusters of infection that would help us potentially in the future to be able to diagnose these infections. And I want to, this is kind of some really exciting data that we're doing, is we now want to actually be able to identify signatures that are specific to these neurologic syndromes. And this becomes very challenging because, as you know, for neurologists, very often you never get kind of a smoking gun in terms of being able to diagnose many of these syndromes. But the idea here is that we took essentially kind of the samples that are the best, most well characterized, the ones that are, say, for instance, brain biopsy confirmed. And we were able to identify signatures for, say, cancer, amyloidosis, MS. It turns out that the classifier actually works really well for multiple sclerosis. I haven't actually looked to see are there EBV-associated pathways, for instance, in this data. Again, this is entirely from looking at gene expression. And I don't really have much time, but I'm just going to end by saying that we're trying to apply this to Lyme disease. So I'm going to quickly go through these slides. But one of the things is that we can use this to generate a classifier that can do Lyme disease diagnosis, potentially with high sensitivity and specificity. We were able to show that not only for Lyme disease, that we can identify gene expression signatures for various types of infections, as you can see here. But the concept is the same, is that can we use host response as a way to identify infections? The last point I want to make, though, is, let me just skip on here, is this, in that by being able to run this assay longitudinally, the idea is that we can not only develop tests that can diagnose infection, but potentially tests that allow us to monitor the course of chronic illness that may be related to the infection. And so this is an example. These are all patients who they have confirmed Lyme disease, presented at time equals 0 with fever and a rash. They were diagnosed with Lyme disease. Most of them are 2-tiered antibody positive. And you can see what I show you here is basically the Lyme disease classifier. And you can see that most of the samples, at least at time 6 weeks or 0 to 6 weeks, they're positive. But you can see that the actual Lyme disease classifier is persistent. So, and we were able to show that it does persist up to 3 months in earlier data, but now we think that it can persist up to 6 months. Now, I haven't unblinded the data, so I don't know which of the severity of symptoms, how many of these patients had persistent symptoms. But you can think that in the future, we'd be able to generate essentially differential classifiers that can distinguish between different types of, that can monitor the course of any given chronic disease. And I just want to end by a few take-home messages. I think that metagenomic next-generation sequencing is very promising, that RNA MNGS data can be leveraged to interrogate a patient's host response. By using this host response profiling using transcriptomics, we can identify biomarkers that will not only allow us to diagnose disease, but allow us to monitor chronic disease. I didn't really mention this, but there's also a lot of interest in looking at the signal from cell-free RNA, which is like looking, for instance, at RNA sequencing of plasma, because then you get the signature coming from dead or dying cells and not the traditional immune response signature coming from immune cells. But that should be considered. And then finally, I want to argue that the development of these types of assays, because it's new technology, it really needs really close coordination with regulatory bodies, including CLIA, CLSI, and the FDA, regarding both the development of reference standards and materials, and perhaps more of a blueprint in terms of how we will validate these assays and get them used in clinical practice. I'd just like to thank all of the members of my lab, in particular the ones highlighted there who are involved in the work, as well as funding from several foundations, the NIH, the CDC, and numerous collaborators. Thank you very much.
No, that's perfectly fine. All right, so.